Hi, I'm Cameron Burke, and welcome to this Kaplan MCAT Science Review for Mechanisms of Enzyme Activity. This video covers how an enzyme gets its job done, beginning with the active site and the methods the enzyme can use to reduce activation energy. We'll also cover binding theories, or the way the enzyme and the substrate bind together. And finally, we'll cover the helper molecules that enzymes need, which are coenzymes and cofactors. The main goal for this review is to be able to describe why enzymes and substrates work so well together based on their chemical compatibilities. By the end of this video, you'll be able to test this goal with practice questions like this one about the ability of enzymes to lower activation energy. Understanding the anatomy of an enzyme is critical to getting the bigger picture of how enzymes work in biological systems. The active site is arguably the most vital piece of the enzyme. This is the spot where substrates stick onto the enzyme. Because enzymes are ultimately just fancy proteins, they're made up of amino acids. The individual amino acids work together to assemble the active site on the enzyme. When the enzyme is in its primary structure, or when it's in a linear arrangement, the amino acid residues that make up the active site can be far away from each other. When the enzyme is in its active tertiary or quaternary structures, the amino acids are folded up and close together, allowing the active site to be formed more tightly. Enzymes are very selective about what substrates can bind to their active site. The active site needs to match a substrate based on a variety of factors that include the shape and size, where the substrate needs to be able to physically fit into the active site, but also includes chemical compatibility factors like polarity and hydrophobicity. One way to illustrate this concept is to compare two relatively similar enzymes like chymotrypsin and trypsin, both of which are involved in protein digestion. In the case of chymotrypsin, serine acts as the active site that binds phenylalanine, activating chymotrypsin so it can cleave the peptide bonds through a hydrolysis reaction in proteins. Trypsin also cleaves, but it manipulates the carboxyl side of amino acids to do so. Because it has a slightly different function, it also has a slightly different binding site, using aspartate to serve as the active site for lysine to bind, activating the enzyme and allowing it to break down proteins as well. Every active site-substrate relationship is a little different, but the job of an enzyme in general is always the same: to lower the activation energy of a reaction. It's important to remember that the free energy of the reaction isn't changed. The enzyme simply makes the reaction require a little less energy to get through the transition state, but it doesn't affect the energy differential between the reactants and the products that causes the reaction to happen in the first place. The enzyme only has one job, which is to lower the activation energy, but there are several ways this job can get done. The first is by stabilizing the transition state. This basically means that the transition state where the reaction begins to go forward exists for longer. One example of stabilizing transition states can be seen in SN2 reactions. The active site binds to the substrate via an SN2 reaction, thus creating a situation where the transition state can be energetically favorable by removing obstacles to stability like torsional strain. The second is through adjusting the microenvironment to make it more favorable chemically, for example by adjusting the pH or keeping water away from the molecule, so water's negative oxygen molecules can't mess with the bonding that should happen for the reaction to be complete. By excluding water from the microenvironment, it stops the extremely polar water molecules from interfering with the forward reaction. The third method of reducing activation energy is to adjust substrate proximity to make sure more of the necessary reactants get close enough to each other. Transient covalent bonding is the fourth way to reduce activation energy for the MCAT. In transient covalent bonding, the substrate binds to several residues in series inside the enzyme, as opposed to just one. There are many steps to transient covalent bonding, but the punchline is that the substrate is vulnerable to a nucleophilic attack, which binds the substrate to the site so the reaction can proceed. The fifth and final mechanism for lowering activation energy to worry about is reactant destabilization. This mechanism basically just works by making the reactants uncomfortable through torsional strain, making the reaction favorable, or by introducing a hydrophobic-hydrophilic interaction that makes the reaction favorable because the molecules are in the wrong hydrophobic state. These 5 methods of reducing the activation energy all help the enzyme accomplish its job of lowering activation energy. We've covered 5 ways the enzyme can help stabilize their transition state, so now we can move on to what happens once the enzyme and substrate have formed a complex. The chemical relationship between the enzyme and the bound substrate is almost solely responsible for the enzyme's ability to get its job done. To initially form the complex, the substrate is added to the enzyme, so the reaction goes forward, and an enzyme-substrate complex is formed. at which point the reaction continues, resulting in a product that the enzyme has disengaged from. Usually, this stops the reaction from going backward as quickly as it initially went forward. So while a little bit of reversal will happen, because the enzyme's not attached anymore, most of the product remains. The specificity of the enzyme-substrate complex accounts for the role enzymes play in reactions. There are two main ideas for what causes the formation of enzyme-substrate complexes. There are the lock and key model and the induced fit model. Of the two models, lock and key is less scientifically accepted. It basically says that every active site fits one substrate, like the way a lock is opened by one key. The enzyme isn't altered until after the substrate binds, according to this model, at which point there can potentially be enzymatic shape changes. This model isn't perfect. There are some things that seem to indicate it doesn't tell the whole story. Competitive inhibition is one of these clues. This is when something that isn't the substrate binds onto the active site, competing with the substrate and preventing the enzyme from working. This contradicts the lock and key hypothesis because if a competitor can bind, then that means there's more than one key that fits into this particular lock. Another piece of weakening evidence is promiscuous reactivity, which is the fact that some enzymes catalyze multiple reactions with different substrates. And if this model were completely accurate, there would need to be a new site for each different substrate. Finally, the lock and key model probably isn't correct because of reverse catalysis. The reaction wouldn't be able to go backward if this model were right, and the Le Chatelier principle says that most reactions can go backward. Unlike the black and white rules of the lock and key model, the induced fit model has some wiggle room. The induced fit model basically says that the active site isn't a perfect fit until the right substrate comes along and makes it one. This is the more scientifically accepted model. Conformational changes are possible before, during, and after the actual reaction, according to this model. Physical changes can happen to both the active site on the enzyme and the substrate, but the end result is that they fit together properly to allow the reaction to go forward. Experimental evidence supports the induced fit model. Competitive inhibition, which we covered a second ago, makes more sense within the induced fit model. And X-ray crystallographs taken of enzymes during reactions help confirm that enzymes take on different forms before and after substrates bind and release from them, by showing the literal protein actually changing formation without a substrate binding to it first. On the MCAT, you need to know about both models, but the induced fit one is the one that you should consider to be more correct. Regardless of the fit model you're considering, some enzymes rely on coenzymes to work. Coenzymes are organic molecules that have to be present for the enzyme to work. Many vitamins are actually coenzymes, which is why they're an essential part of your diet. Adenine, as well as vitamins C and A, among others, are all examples of coenzymes. Vitamins in particular you might want to be familiar with on test day include A, B, C, D, E, and K. Vitamins are either fat-soluble or water-soluble. Since B and C are the only two water-soluble vitamin groups, it's easiest just to remember that those two are water-soluble, and know that any other vitamin on the MCAT is going to be fat-soluble. When coenzymes are bound on super tightly, they're called a prosthetic group because they're basically just part of the enzyme. Cysteine residues on heme C are one example of a prosthetic group, because although cysteine isn't technically part of heme C can't work without cysteine, and the two are so tightly bound together that they function as a single molecule. Coenzymes are super important organic molecules that enzymes need, but sometimes enzymes need inorganic molecules too. The inorganic ones are called cofactors, but they basically have the same job as a coenzyme. Usually cofactors are metal ions. When an enzyme has metal ions bound to it, it's referred to as a metalloprotein. There are a lot of minerals people need in their diets to make all of their proteins run smoothly, which is why there are so many in grain cereals and foods otherwise devoid of nutrients that have minerals artificially added to them. If all the cofactors and coenzymes an enzyme needs are there, it's called a holoenzyme. This means the enzyme is whole and complete so it can work. If the enzyme doesn't have all the extra stuff it needs, it's an apoenzyme, and it can't get its job done. Their function is basically the same, but on test day it's important to remember that organic helpers are called coenzymes, while inorganic metal helpers are cofactors. Now you can check out how you're doing with regards to the learning goals. Check out this question about enzyme fit models. The correct answer is A. If movement is happening with no substrate at the site in question, it weakens the idea of the lock and key model. Next, try this question about activation energy. C is the right answer. If the transition state isn't stabilized, raising the energy of the reactants, perhaps by putting strain on them, is the next best step. The final practice question deals with cofactors. B is correct. Because most cofactors are metal cations, it makes sense to have a negative charge on their binding site. To recap, there are 5 ways enzymes can catalyze a reaction that you should review for test day, all of which reduce activation energy. Regardless of the catalyzation method, there are 2 binding models to care about: lock and key and induced fit. But lock and key isn't as good as induced fit, so focus on induced fit for test day because it has more scientific support. Finally, enzymes need helpers, and those helpers come in the form of both vitamin coenzymes and mineral cofactors. Thanks so much for watching this Kaplan MCAT Science Review for Mechanisms of Enzyme Activity, and good luck when you take your test!